Hi class, this is Dr. Cassandra Quave, and in this lecture we'll be covering Chapter 8 from your textbook, discussing gene transfer in genetic engineering. Now, the transfer of genetic material from one organism into another can have huge consequences that may be far-reaching. In microbes, it provides a way for viruses to introduce genetic information into bacteria and mechanisms for bacteria to increase their disease-causing capabilities or to become resistant to antibiotics. Information obtained from the study of transfer of genetic material between microbes has many different potential applications in industries including agriculture and the medicine and pharmaceuticals. And it also can be used to help solve some of the problems associated with prevention and treatment of infectious disease. Here on this first slide you can see a picture of one of the most famous geneticists associated with the understanding and these large projects involving gene transfer and genetic engineering. This is Dr. Venter. And if you look inside your module for this lecture, you will also see a link to a lecture given by Dr. Ventner on some of his Institute's initiatives in genetic information that I think you'll find quite interesting. Now let's go over some basics with regards to gene transfer. First, let's define gene transfer. Gene transfer basically refers to the movement of genetic information between organisms. Now, in most eukaryotes, it's an essential part of the organism's life cycle and typically occurs through sexual reproduction, in which male and female parents produce gametes or sex cells, which, when united, can form a zygote, or the first cell of a new individual. Because each parent produces many genetically different gametes, there are many different potential combinations of the genetic material that may be transferred to the offspring. However, in bacteria, which are prokaryotes, gene transfer is not an essential part of the life cycle. When it does take place, however, usually only some of the genes of the donor cell are transferred to the other cell, known as the recipient cell. And the combination of genes, or DNA, from two different cells is called recombination, and the resulting cell is referred to as a recombinant. Now, eukaryotes again participate in something known as vertical gene transfer, and this is when genes pass from parents to their offspring. Now, sexual reproduction of plants and animals is what we usually think of as vertical gene transfer. However, in contrast, again, bacteria can do vertical gene transfer when they reproduce asexually by binary fission. Furthermore, bacteria can also do horizontal or lateral gene transfer, in which they pass genes to other microbes of their same generation. Gene transfer is a significant part of prokaryotic life because it greatly increases the genetic diversity of organisms for prokaryotes and eukaryotes, actually. Now, mutations can account for some genetic diversity, but again, gene transfer between organisms accounts for even more. Now, some of the advantages of genetic diversity are that when organisms are subjected to changing environmental conditions, genetic diversity can actually act to improve their likelihood that some of the organisms will actually be able to adapt to that particular condition. And this diversity leads to evolutionary changes. Organisms with genes that allow them to adapt to an environment to survive and reproduce, whereas other organisms that may lack these genes will perish. If all organisms were genetically identical, all would survive and reproduce, or all would die. And this concept trickles down to other important areas in which we use genetic engineering, such as in crops, where you have monocrops being used, or plants of the same lineage. If they then are attacked by some pathogen and they don't have that genetic diversity, the crops could be in great danger. Now, in recombinant DNA technology, genes from one kind of organism can actually be introduced into another kind of organism using lateral transfer. An example of this would be when human genes are inserted into the cells of a pig. Genetic engineers have learned to artificially manipulate the three natural means of lateral gene transfer to create the desired recombinant DNA and the organisms. Let's go over these in the next few minutes. Let's go over some of these three basic mechanisms now. The first of these is transformation. Bacterial transformation is a change in an organism's characteristics because of the transfer of genetic information, and this was first discovered by Frederick Griffith in the 1920s during his studies on pneumococcal infections in mice. Griffith's study is still one of the best known today for transformation. Here you can see in the figure that the setup of his experiment. Now, pneumococci with capsules produce a smooth type, glistening colonies, and those lacking capsules produce rough type colonies with a coarse, non-glistening appearance. Only the capsule producing pneumococci inoculated into mice were actually the pathogens. They were the only ones that could cause pneumonia in the mice. Mice are found to be very sensitive to pneumococci, and so they make a really good test because of the fact that the mouse would then die if exposed to this smooth type capsule. Now, in Griffith's experiment, he injected one group of mice with heat-killed smooth pneumococci, shown here in the far left. He then injected a second group with live smooth pneumococci, and you can see that they died. Then he also, in a third group, he injected mice with live rough pneumococci. These were the ones lacking capsules, and they lived. Now, what made his experiment really interesting is that in a fourth group, he injected a mixture of live rough type capsules and the heat-killed smooth type he injected a mixture of live R-type or rough type colonies with heat-killed smooth type colonies. Those are the ones that had capsules but that had been killed. And again, neither of these were lethal by themselves, as you can see in the top left and right part of your diagram, but when combined, they caused the mice to die. What makes this even more interesting is that then, after death, the live smooth type organisms, as well as the R types, were recovered from the dead animals. We now know that the rough type bacteria were actually able to pick up the naked DNA that was liberated from the disintegrated dead smooth type bacteria, and they were able to incorporate this into their own DNA. So the rough type bacteria that picked up the smooth type DNA then had genes for capsule production and became genetically transformed into smooth type organisms, making them lethal in this particular experiment. Now, to understand the mechanism of transformation, scientists can extract DNA from donor organisms through complex biochemical processes that yields hundreds of naked DNA fragments from each chromosome. When this naked DNA is placed in a medium with organisms capable of incorporating it, most organisms can take up a maximum of about 10 fragments, which is less than 5% of the amount of DNA normally present in the organism. Now, uptake of DNA only occurs at a certain stage in a cell's growth cycle in response to high cell density and depletion of nutrients. In this stage, a protein called the competence factor is released into the medium and helps to facilitate the entry of DNA. When competence factor from one culture is used to treat a culture that lacks it, cells in the treated culture become competent to receive DNA, meaning that they can now take up DNA fragments. And here we can see the mechanism of bacterial transformation in your figure. Here you have naked DNA fragments from the disintegrated cells in part 1 of the diagram. These must be of the correct genus and be in the state of competence, or the proper physiological condition to permit the entry of those fragments. In the next stage, some of these fragments enter into the competent cell. In the third stage, they go through recombination, and DNA that is not recombined into the host cell is then broken down by enzymes. Now, although transformation has been observed mainly in the lab, it does also occur in nature, and it probably follows what happens during the breakdown of dead organisms in an environment where the live ones of the same or closely related species are present. However, the degree to which transformation contributes to the genetic diversity of organisms in nature is still not fully understood. Now let's cover one of the second basic mechanism for lateral gene transfer, and this is that of transduction. Now, in order to understand transduction, you first need to understand some of the basic properties of bacteriophages, also known as phages. Phages will be discussed in greater detail in a later chapter, but the basic thing that you need to know is that they're basically a core of nucleic acid covered by a protein coat, and these are a kind of virus that infects bacterial host. It will then reproduce inside of them, shown here in your figure. A phage capable of infecting a bacterium will attach to a receptor site on the cell wall. The phage nucleic acid will then enter into the bacterial cell after a phage enzyme weakens the cell wall. The protein coat remains outside, attached to the cell wall. Once the nucleic acid is in the cell, further events follow one of two pathways, and this depends on whether the phage is determined to be virulent or temperate. And you can see more about this lytic and lysogenic life cycle on page 217 of your book. Now, a virulent phage is capable of causing infection and eventually the destruction and death of the bacterial cell. The temperate phage, on the other hand, does not cause a disruptive infection. Instead, the phage DNA is incorporated into the bacterium's DNA and is replicated with it. Phage DNA that is incorporated into the host bacterium's DNA is called prophage. Now, persistence of a prophage without phage replication and destruction of the bacterial cell is known as lysogeny, and cells containing enough prophage are said to be lysogenic. This is in contrast again to the lytic cycle, in which phage can actually destroy the host cell. Now, temperate phages can replicate themselves either as a prophage in the chromosome or independently by assembling into new phages. And transduction happens when instead of only phage DNA being packed into newly formed phage heads, some of the bacterial DNA can also be packed into the heads. Temperate phages can carry out both generalized and specialized forms of transduction. In generalized transduction, any bacterial gene can be transferred by the phage, but in specialized transduction, only specific genes are transferred. Now, let's discuss more about specialized transduction. There are several lysogenic phages that are known to carry out specialized transduction, but there's one in particular known as the lambda-phage in E. coli that's been studied the most. Phages usually insert at a specific location when they integrate with a chromosome. Lambda-phage inserts into the E. coli chromosome between the gal gene, which controls galactose use, and the bio gene, which controls biotin synthesis. And these are both part of operons. So when cells containing lambda-phage are induced to enter the lytic cycle, genes of the phage form a loop and are excised from the bacterial chromosome. The lambda-phage DNA then directs the synthesis and assembly of new phage particles, and the cell will break up or lyse. Now, when this phage then infects another bacterial cell, the particle transfers not only the phage genes, but also that gal gene. And this process in which a phage particle transduces or transfers specific genes, again, from one bacterial cell to another, is called specialized transduction. This is in contrast with generalized transduction, in which a bacterial cell containing phage DNA enters into the lytic cycle. The phage enzymes break open the host cell DNA into many segments, and the phage is packaged randomly with different parts of this DNA. This figure illustrates generalized transduction. Here you can see a bacteriophage infection of a host bacterium will initiate the lytic cycle, and the bacterial chromosome is broken into many fragments, and any of these can be picked up and packaged along with phage DNA into new phage particles. And those particles are released and infect another bacterial cell, and at this time the new host acquires the genes that were brought along or transduced from the previous bacterial host. Now, this process of transduction is really important for many reasons. First is that it can transfer genetic material from one bacterial cell to another and can alter the genetic characteristics of the recipient cell. Also, the incorporation of phage DNA into a bacterial chromosome demonstrates a close evolutionary relationship between prophage and the host bacterial cell. Third, the discovery that a prophage can exist in a cell for a long period of time suggests a similar possible mechanism for the viral origin of cancer. If a prophage can exist in a bacterial cell and at some point alter expression of the cell's DNA, this could explain how animal viruses cause malignant changes. Another interesting thought is that some animal viruses probably brought along genes from their previous hosts when they infected new human hosts. And lastly, and this is most important to molecular genetics, is that transduction provides scientists with a way to study gene linkages. Genes are said to be linked when they are so close together on a DNA segment that they are likely to be transferred together. So different phages can be incorporated into a bacterial chromosome, with each kind usually entering at a specific site. Thus, by studying many different phage transductions, scientists can determine where they are inserted on the chromosome and which adjacent genes they are capable of transferring. The combined findings of many studies can eventually show identification of a sequence of genes in a chromosome, and this procedure is called chromosome mapping. Now, a third important mechanism of lateral gene transfer is that of conjugation. In conjugation, much like transformation and transduction, again, genetic information is transferred from one bacterial cell to another. However, conjugation also differs from these two previous procedures in two ways. It requires contact between the donor and the recipient cell, and it transfers much larger quantities of DNA, and it can occasionally even transfer entire chromosomes. Conjugation was first discovered by Josh Lederberg in the 1940s. He used mutated strains of E. coli that were unable to synthesize certain substances, and he selected two strains, each of which were defective in a different synthetic pathway, and grew them up in nutrient-rich medium. He removed the cells from each culture and washed them to remove the residue of the medium, and then attempted to culture cells of each strain on agar plates that lacked the nutrients needed by the strain. He also mixed the cells from the two strains and plated them on the same medium. Whereas cells from the original cultures failed to grow, some of the mixed cultures did grow. Now, this worked, of course, because of conjugation, and here you can see a figure from your book that explains the transfer of F plasmids. Here you can see an F plus and an F minus cell. The F plus is for the donor cell, F minus is for the recipient cell. The F plus cell will transfer one strand of DNA from its F plasmid to the F minus cell via the conjugation bridge. And as this occurs, the complementary strands of F plasmid are synthesized. and so the recipient cell gets a complete copy of the F plasmid, and the donor cell still retains a complete copy. Now, this pathway of F plasmid transfer has been studied extensively, and other studies have found that there are actually high frequency strains, or strains in which donor strains which arise from F plus strains when the F plasmid is incorporated into the bacterial chromosome at one or more possible sites. So these high frequency of recombination, or HFR strains, tend to have a higher frequency of recombining through this pathway. And this is again due to their location on the chromosome. Now, conjugation is a highly significant aspect of gene transfer because it contributes again to genetic variation, and the fact that larger amounts of DNA are transferred in conjugation than in other transfers. So conjugation is very important in increasing genetic diversity. Now, plasmids that are self-transmissible or have genes for the formation of an F pilus can sometimes even transfer into species other than their own kind, and these are said to be promiscuous. Sometimes the species are only distantly related. In other cases, a transfer may even occur into eukaryotic cells. This obviously has important implications for health and evolution. Now, before we move on to plasmid types, I just wanted to reiterate this aspect, that the most fundamental differences among those major types of lateral transfer that we've discussed so far, and again, just to revisit, these included transfer through transformation, transduction, and conjugation. The most significant difference is the quantity of DNA transferred and the mechanism by which the transfer occurs. In transformation, less than 1% of the DNA in one bacterial cell is transferred to another, and the transfer involves only chromosomal DNA. In transduction, the quantity of DNA transferred varies from a few genes to large fragments of the chromosome, and a bacteriophage is always involved in this transfer. In specialized transduction, the phage inserts into the bacterial chromosome and carries out a few host genes with it when it separates. But in generalized transduction, the phage causes fragmentation of the bacterial chromosome, and some of the fragments are accidentally packed into viruses as they are assembled. In conjugation, the quantity of DNA transferred is highly variable and depends on the mechanism. And here, a plasmid is always involved in the transfer. An F plasmid itself can be transferred, as occurred in F plus and F minus conjugation. An initiating segment of a plasmid and any quantity of chromosomal DNA, ranging from a few genes to a whole chromosome, can be transferred through high-frequency conjugation. A plasmid and whatever chromosomal genes it has carried with it from the chromosome are transferred in F prime conjugation. And you can, again, look at these in page 223 of your textbook and table 8.2. It has a nice summary of some of the similarities and differences between these. Now, moving on to plasmid types, let's discuss the fertility plasmid, resistance plasmids, and bacteriocin plasmids. F plasmids, or fertility factors, direct the synthesis of proteins that self-assemble into conjugation pili. Resistance plasmids, or R plasmids, carry genes that provide resistance to various antibiotics such as chloramphenicol and tetracycline, and to heavy metals like arsenic and mercury. Now, other plasmids that direct the synthesis of bactericidal or bacteria-killing proteins called bacteriocins. Now, a typical resistance plasmid can carry genes for resistance to various antibiotics and to inorganic toxic substances, sometimes used in disinfectants. This is shown here in the figure. The resistance transfer factor includes genes needed for the plasmid to undergo conjugation. Now, most plasmids are circular, double-stranded extrachromosomal DNA. They're typically self-replicating by the same mechanism that any other DNA uses to replicate itself, and most plasmids have been identified by virtue of some recognizable function that they serve in a bacterium. Other plasmid types include virulence plasmids. These include those such as in Salmonella, or the neurotoxin genes carried on plasmids found in Clostridium tetani, which cause disease signs and symptoms. Tumor-inducing plasmids can cause tumor formation. This is limited to plants. And whereas other plasmids may contain genes for catabolic enzymes, and generally plasmids carry genes that code for functions not essential for cell growth. The chromosome carries the genes that code for essential functions. Now, in addition to being transferred on resistance plasmids by conjugation, your R genes, or resistance genes, can also move from plasmid to another cell or even become inserted into the chromosome. And the ability of a genetic sequence to move from one location to another is called transposition. And such a mobile genetic element sequence is called a transposable element. Transposable elements replicate only when in a plasmid or in a chromosome. During transposition, the insertion sequence is copied by the transposase and cellular enzymes. The copy randomly inserts itself into the bacterial element and remains in its original position. However, the ability to move among plasmids or to a chromosome greatly increases the ways in which transposable elements can affect the genetic makeup of a cell. A transposon is a transposable element that contains genes for transposition, and one or more other genes as well. Typically, these are genes for toxin production or are resistance genes. Thus, a transposon can be used to move resistance genes from one plasmid to another or to the bacterial chromosome. Viruses and plasmids can even move the transposon to a different cell, even to one of a different species. This movement can also occur between prokaryotes and eukaryotes. Here you can see the figure concerning transposons. Here, a typical transposon is bounded by inverted repeat terminals, or DNA segments with base sequences that are identical when read in opposite directions on different strands. A gene coding for a transposase enzyme that cuts DNA at these insertion sequences allows the transposon to cut itself out into and out of plasmids and chromosomes. There is also a gene for a repressor protein that can keep the transposase gene from being transcribed. Now, in 1983, Barbara McClintock won the Nobel Prize for her work on transposons using corn. Transposons were next found in microorganisms and are now considered a universal phenomenon. Transposition is a relatively rare event and not easily detected in eukaryotes. It is easier to detect in bacteria because researchers can work with a large population that can be tested more easily for particular characteristics. Now let's move on to a discussion of more about genetic engineering and genetic fusion. Now, genetic engineering refers to the purposeful manipulation of genetic material to alter the characteristics of an organism in a desired way. Various methods of genetic manipulation enable microbial geneticists to create new combinations of genetic material in microbes. The transfer of genes between different members of the same species occurs in nature and has been done in the laboratory for several decades. Here we're going to discuss 5 techniques of genetic engineering, including genetic fusion, protoplast fusion, gene amplification, recombinant DNA technology, and the creation of hybridomas. Let's begin with genetic fusion. Genetic fusion allows the transposition of genes from one location on a chromosome to another. It can involve deletion of a DNA segment, resulting in coupling of portions of two operons. Here you can see a figure concerning genetic fusion. One possible example in which deletion of part of a chromosome will cause two different adjacent operons to be joined together. The control mechanisms of the first operon will now govern the expression of the genes that were originally part of the second operon. One way that genetic fusion has been used in genetic engineering is this example of Pseudomonas syringae, which is a bacterium that grows on plants. Genetically altered strains have been developed that increase the resistance of plants like potatoes and strawberries to frost damage. Strains of this bacterium, which occur naturally on the leaves of plants, produce a protein that forms a nucleus for the formation of ice crystals. The ice crystals can damage the plants by causing cracks in the cells and leaves. By removing part of the gene that produces the ice crystal protein, scientists have engineered strains of this bacterium that cannot make the protein. When the organisms of this strain are sprayed onto the leaves of the plants, they crowd out the naturally occurring strain, and the treated plants then become resistant to frost damage at temperatures as low as -5 degrees Celsius. Moving on to another form of genetic engineering is protoplast fusion. A protoplast is an organism with its cell wall removed, and protoplast fusion is accomplished by enzymatically removing the cell walls of organisms of two strains and mixing the resulting protoplasts. This allows for fusion of the cells and their genetic material, allowing specifically that material from one strain to recombine with that from the other strain before new cell walls are produced. Although genetic recombination occurs in nature about 1 in a million cells, it occurs in protoplast fusion in as many as 1 in 5 cells. So this is a way of simply speeding up a process that occurs in a very limited way in nature. Protoplast fusion has been used to fuse bacterial, fungal, and plant cells. It can be used again to mix two strains, each of which has desirable characteristics, and the new strains with both characteristics can then be produced. It works best in strains of the same species. It has been accomplished in molds, however, even between two species of the same genus, and in yeast among two different genera. Now let's discuss gene amplification. Gene amplification is a process by which plasmids, or in some cases even bacteriophages, are induced to reproduce within cells at a rapid rate. If the genes required for the production of a substance are in the plasmids or can be moved to them, increasing the number of plasmids will increase production of the substance by the host cells. Most bacteria and many fungi, including those that produce antibiotics, contain plasmids. And these plasmids often carry genes for antibiotic synthesis and can provide many opportunities for using gene amplification to increase antibiotic yields. The possible applications of gene amplification are not limited to increasing antibiotic production, though. In fact, it may be even more effective in increasing production of substances that are synthesized by simpler pathways, such as substances like enzymes and other products like amino acids, vitamins, and nucleotides. Now, with regards to recombinant DNA technology, this is one of the most useful of all techniques of genetic engineering. Now, recombinant DNA is DNA that contains information from two different species of organisms. If these genes integrate permanently into the egg or sperm cell so that the genes can be transferred to offspring, the resulting organism is said to be transgenic or recombinant. Making recombinant DNA involves three processes. The first is manipulation of DNA in vitro, that means outside cells. The second is the recombination of another organism's DNA with the bacterial DNA in a phage or plasmid. And third is the cloning or production of many genetically identical progeny or of phages or plasmids that carry the foreign DNA. Let's go over this figure together that illustrates how to go about producing recombinant DNA. Here, the first step is to obtain the release of DNA containing a gene of interest from mammalian or other cell types. The second step involves construction of recombinant DNA with DNA molecules with the use of restriction enzymes like endonucleases that can cleave DNA at specific sites and into short segments. Cleaving the DNA leaves sticky ends that can be joined by hydrogen bonding to similar ends on any DNA segment, cleaved with the same endonuclease. In the third step, the recombinant DNA is introduced to a host bacterium. In the fourth step, cells that carry the gene of interest are identified and isolated, and cloning vectors, like plasmids, can carry one or more genes for antibiotic resistance, which are of value in isolating and identifying cells that carry the recombinant DNA. Now, one of the most medically significant applications of recombinant DNA technology is the modification of bacterial cells to make substances that are useful to humans. So, for example, in order to make bacterial cells produce human proteins, a human DNA gene with the information for synthesizing that protein can be inserted into the vector. An example of this is interferon, which is a substance used to treat certain viral infections and cancers. Also, the hormone insulin was one of the first products made through this technology. Human growth hormone can also be made this way, and new products like vaccines, blood coagulation proteins, and enzymes like cholesterol oxidase can now be produced in larger quantities using this technology. Now, recombinant DNA is also being used to make vaccines more economically and in larger quantities than ever before. Genes that direct the synthesis of specific substances called antigens from a disease-causing bacterium, virus, or protozoan parasite can be inserted into another organism, and then this organism makes a pure antigen. When the antigen is introduced into a human, the human immune system makes another specific substance called the antibody, which takes part in the body's defense against the disease-causing organism. Now, there are also other kinds of applications, such as those involved in industry and in agriculture. For example, in industrial applications, fermentation processes used in making wine, antibiotics, and other substances could be greatly improved by the use of recombinant DNA. For example, addition of genes for the synthesis of amylase to the yeast Saccharomyces could allow these organisms to produce alcohol from starch. As far as agricultural applications, some bacteria are being engineered to control insects that destroy crops. There are many different potential directions that can be taken through these technologies. However, many of these can also lead to certain ethical concerns, which are discussed in your chapter. I want to wrap up with a discussion of hybridomas. So along with the study of genetic recombinations in microbes, also came studies of combinations in higher organisms. Here you can see a picture of a hybridoma cell. This is a single cell fused from two other cells, and these are often made by fusing an antibody-producing plasma cell and a cancer cell. The latter keeps the culture dividing and growing indefinitely, but the former causes the hybridoma to produce pure antibody against the antigen to which it was sensitized. Hybridomas can be grown in the laboratory and can be used to produce pure specific antibodies called monoclonal antibodies against any antigen to which the white blood cell was previously sensitized. Prior to the production of hybridomas and their monoclonal antibodies, there was no source of pure antibodies that existed. Now we have many different kinds of monoclonal antibodies that are produced commercially, and these represent a huge step forward in the field of immunology. Some of the risks and benefits of working with recombinant DNA could be some of its potential hazards. There were early fears that recombinants could prove to be new and especially virulent pathogens that could wreak havoc upon humans and who would have no natural defenses or treatments against it. In fact, in the 1970s, there was a moratorium called on certain experiments until hazards of this technology could be really assessed. From this assessment came the idea of biological containment, or the practice of making recombinant DNA only in organisms that have mutations that would prevent them from surviving outside of the laboratory. However, in the 1980s, some of the constraints on this technology were relaxed because no illness in laboratory workers could have been traced to recombinants. The strains that were being used in experiments did not typically infect humans, and incorporation of mammalian genes into E. coli was observed in nature, and these genes invariably impaired the organism's infectious or ability to adapt to the environment, and so on. And most scientists now agree that recombinant DNA techniques do offer significant benefits and exceedingly small risks to humans when they're under study. This is all I have for today's chapter. Please make sure that you have read your chapter carefully, gone through your review, and done the practice questions and your review sheet provided within Blackboard. Thank you.